it's just saying that it's glucose alpha 1,2 fructose, but that's technically incorrect, right? Because that should be an alpha 1 beta 2.
It should be an
For sucrose.
oh, sucrose.
Yeah.
Sucrose is, here's your anomeric, right? So they're opposite, they're trans, that's beta. They're opposite. I'm sorry, that's alpha.
Yeah,
Trans is alpha.
Yeah.
They're opposite, trans, it's alpha.
I thought this what the anomeric carbon is here, and then it's the last one, so it's this one that's beta.
Let me see. This is the anomeric.
Yes, and then you count the
Oh yeah,
last.
you're right. That's beta.
Yeah.
That's right.
So that's
beta.
alpha.
That's alpha 1 beta
2.
Beta 2. Yes.
So
Alpha
really
1
it
beta
should be alpha
1 beta 2.
Fructose.
What do they have?
They have glucose alpha 1 2
fructose. Beta.
So they really
Beta.
okay. Looks like in this book they only count like the first bond.
But you should only count the bond if it's the glycosidic that's participating, if it's the anomeric participating in the glycosidic, and it is.
It is, yeah.
So it should be named.
So really it should be.
It should be named.
Yeah.
They're just not naming it.
Okay.
They're not saying it's alpha.
Yeah.
They're just not naming it. But it needs to be named because that's the anomeric and it's participating.
Yeah. Other question was NADH and FAD are both organic, so they would be referred to as coenzymes technically, right?
Yes.
Okay. Well,
They're cofactors, which
cofactors.
they are cofactors, and a subcategory of cofactors are.
Okay.
So they're not wrong, but they're just not specific.
Okay.
If that makes sense.
Yeah, sure. Thank you.
Yeah. Okay. How we doing? Let's see. In my chat. What else do I need today? I think that's it. We're on section 2. Right? Good morning. Y'all have a good happy Halloween. Halloween did freak me out a little bit because nobody was wearing masks. It freaked me out a little bit. I was like, Charles, you stay at home. You can sit with us. When we sit like 8 feet back from the table that has all the candy, and we're good. Okay, so let me open up attendance. It was really super busy. Oh. All right, so go ahead and click in on your attendance when you get a chance. I don't think I'd want to work in the ER during Halloween. I mean, that just sounds scary.
That's what you want to do?
Just sounds scary.
Yeah. What are you talking about? Yeah. What are you talking about? Yeah, bro. What are you talking about?
Exactly what he said. Did he have to say it again?
I was drawing one on time.
Okay, so our exam is on Friday. Exam is on Friday. So I posted, I didn't know how far we would get through 10.3 today. So what I did was I made the 10.3
What are you talking about?
section as a whole video. So you can pick up anywhere in it. And then 4 and 5 are like super insanely short. So, you know, not a big deal. Make sure everybody had everything.
I usually go on campus for class, not to Jordan and Corey's apartment.
Okay, so today we are going to talk about what happens to pyruvate
Let's take a logical guess where I
after
might.
glycolysis. I'm going to start off with a joke, right? So, so why did the moon have a upset?
Should I ask Dr. B right now? She'll know.
It was a full moon. I thought that was kind of good for Halloween, right? Around Halloween. You guess so. I work for this stuff, and you know. All right, so don't forget to do attendance before I don't know, before the end of class. Yes, ma'am.
Sorry, it's a silly question.
Go ahead.
What section is this?
10.2.
No, like the course 4M, 4.
Oh, 4M.
4,
4M. Yes, 4M. Yeah. Okay. So we have to, before we can actually start the citrate cycle, we have to convert pyruvate to acetyl-CoA, right? Because glycolysis, we got pyruvate. Well, that's handy dandy. But in order to start the citrate cycle, we have to have acetyl-CoA. So we have to transport pyruvate from the cytosol into the mitochondrial matrix, right? Now, once it's inside the matrix, then we can convert it to acetyl-CoA and we can go ahead and start all that oxidation reduction fun. So in order to do that, you have one enzyme that completes the process. It's called pyruvate dehydrogenase. Now, pyruvate dehydrogenase, we say it's one enzyme, but it's really a complex. It's really quite massive. There are three main subunits, and we name them E1, E2, and E3. And then, in addition to, these are the protein subunits, you also have coenzymes. Now, these coenzymes are super important. Usually, right, if you have an enzyme, it's those amino acid side chains that are going to participate in the reaction mechanism, right? But really, it's limited what those amino acid side chains can do. And so the more coenzymes you have, basically the more that you can do. And redox reactions typically require multiple coenzymes. All right, so let's take a look at how that works. So we know, I want this to fit all in one, but it doesn't right now. I don't know. Let's see. Okay, well, so we know that we're going to use NAD plus and we're going to use FAD, right? We talked about that last time. These are the two molecules that are going to participate in that redox reaction, right? Redox is all about moving what? Moving electrons. So these are really common. That shouldn't be enzymes, that should be coenzymes in oxidation reduction reactions. So you probably know a lot of the coenzymes that are going to participate in pyruvate dehydrogenase because they are the B vitamins, right? You've seen that at the store, your B vitamin complex and all that, right? Okay, well, now you're going to know what they're really for. So NAD is derived from niacin, or is sometimes called B3. FAD is derived from riboflavin, or vitamin B2. So NAD tends to participate in catabolic reactions. CATA, catabolic reactions. Whereas NADP, have y'all heard NADP before? It's a similar carrier, but this is anabolic. And it's kind of one of the ways that the cell can regulate whether you're going to go anabolic or catabolic. Catabolic is NAD. Catabolic is NAD. Anabolic is NADP. Y'all know those P's, right? All right, and this is not the whole structure of NAD. Do you see this R right here? This R is telling you got a bunch of other stuff going on, right? So anybody know what NAD stands for? Nicotinamide, adenine, dinucleotide. So this is basically the nicotinamide, and then you've got a dinucleotide attached to it, but we're not showing it. We're just showing the important bits, right? So when we look at this, what's going to happen is, here's our oxidized substance, NAD+. When we undergo that oxidation reduction reaction and we form a reduced version of this molecule, if we're going to reduce, what's going to happen to hydrogens? We're going to gain hydrogens. So look at this carbon right here. It's participating in a double bond on this side, single bond on this side. Now what happened? We picked up a hydrogen on here, and then we also have another hydrogen. It's not free. I know it looks like you have it on one side and nothing happened to it, but it actually comes from the same molecule, right? You're going to pull hydrogens off of one molecule and you're going to stick one on the NAD plus, and then you're going to have one that's in the mitochondrial matrix. Okay. So FAD, similar sort of deal, right? We have a big old molecule, but we're not going to worry about too much of it. We're just kind of going to worry about this nitrogen right here and this nitrogen right here. Remember how we said it was a two-step mechanism when we're going to FAD? So in the first step, one of these nitrogens is going to pick up a hydrogen. It also picks up an electron at this carbon next to the nitrogen, where we lost that double bond. See, between the nitrogen and the carbon of the double bond, the nitrogen gets a hydrogen and the carbon gets an electron.
So wait, does the FAD lose or gain electrons?
When you go from FAD to FADH2, you are gaining hydrogens and electrons.
Okay.
So we're showing these both going from oxidized to reduced. Okay, so our nitrogen picks up a hydrogen and an electron. Here our nitrogen still has that hydrogen. Now look at the other nitrogen. Our nitrogen picked up a second hydrogen, and then we have also picked up that second electron. So we just showed this because this is a very, very unstable intermediate, right? It doesn't hang out in this form for very long. It immediately converts over to this one.
So it gains electrons and hydrogen.
Yes, at each stage, it gains a hydrogen and an electron, a hydrogen and an electron. Okay. So you need to eat these in order to survive. That's what a vitamin is. So these were originally identified because when you had deficiencies of these in your diet, you would have some kind of physical symptom. So lots of reactions inside of your body are going to use NAD plus and FAD. And so if you don't have these, you're going to have particular symptoms depending on what the vitamin it is that you are missing. And so they were named before we really understood what role they had. So that's why they have two names. Okay, so don't let that throw you off. All right, coenzyme A. What are we making? What do we have to have in order to enter glycolysis? Not glycolysis, citrate cycle. Acetyl-CoA. What's CoA? CoA is coenzyme A. This is derived from vitamin B5. Panthenoic acid is another name for it. You don't have to know that though. But this is another one that's essential for life. So this is what we use to generate that acetyl-CoA. So this whole thing is coenzyme A. Okay. At the very end of coenzyme A, you have a very reactive thiol group. That thiol group is going to pick up an acetyl group that comes from pyruvate. So this right here is from pyruvate. Now you have what you need in order to begin the citrate cycle. So this thioester bond that's formed has a lot of energy inside of it, right? And so it makes it really an ideal carrier compound for that acetyl group. Because what we're going to see is we're going to see those carbons kind of being passed around, right? That's the whole name of the game, really, passing the carbons. All right. Thymine pyrophosphate, they call it TPP. This is another B vitamin, right? And this is all part of what? As we're naming all these things, what are all these things? So this is all the pyruvate dehydrogenase complex. So we're talking about all the coenzymes that are involved in that, right? The coenzymes, good, okay. So TPP is required for converting our aldehyde transfer. So watch, let's see. I can move this. All right, so right here on this carbon, see the nitrogen that has a partial positive charge? The carbon that's double bound attached to it, this is an attachment site for the activated aldehyde. So remember I said you're passing the carbons? The next place that carbon is going to be passed to is right there. No, the carbon right here, the carbon right there.
So that's where the carbon's going to be.
That's where you're going to attach, that's where you're going to attach the carbons on. The aldehyde groups will attach right there. We're going to look at the whole mechanism, but I just kind of want to introduce each of the individuals, and then we're going to look at the whole thing. We got one more.
So it's so where like the carbon underneath the input.
Below the nitrogen, yeah, below the nitrogen. Okay. All right, so the last one is alpha lipoic acid. So this one is really kind of cool because right here we have this reactive disulfide. So the reactive disulfide is right here. This is going to participate in our redox reactions, right? And so just like we said, we're passing the carbons. What did we just pass? What kind of group is that? It is an aldehyde, but it's an acetyl group. The red, in red is the acetyl. Right? Acetyl is a methyl, single bonded to a double bonded, a carbon double bonded to an oxygen. That's an acetyl group. Yeah? No? Yes. Okay, good. All right. So you're gonna transfer it from TPP. So TPP is going to have that acetyl group, and then it's going to transfer away from TPP. So TPP is free, and it's going to bind onto the lipoamine, and then now we'll have acetyl dihydrolipoamine. But it's going to make more sense when we look at the whole thing all together. Yep, it comes from TPP. So the acetyl group was in hydroxyethyl TPP. And then it gets transferred off of TPP onto our acetyl dihydrolipoamide. Okay, so let's look at the whole thing. All right, and you just got to follow the arrows on this. So this is our whole complex. This is pyruvate dehydrogenase, the whole thing. We have our E1, our E2, and our E3 protein subunits, right? So when you come in, what do you look like? Your pyruvate, how many carbons do you have? 1, 2, 3 carbons, right? Yes? Okay, so we got 3 carbons. So the first thing that's going to happen is that we're going to remove one of those carbons as CO2. So it's that E1 subunit, right? We're right here. This is what's going to happen. We're going to have one of the carbons come off as carbon dioxide, and now we're going to have 2 carbons. So those 2 carbons are going to attach to TPP. Once we have it attached to TPP, now we're going to go this way, right? And if I want to follow, let's follow the red stuff first. Right? Where do those 2 carbons go? Remember we said we swapped them from TPP to the alpha lipoamine, right? So here they are. But at the same time, what have you done? You've regenerated TPP to go again, right? Now, in the next step, right, in E2, we're going to convert, we have that, what used to be a disulfide bond, we broke that disulfide bond in order to attach our acetyl group. Now what we want to do is move that acetyl group to CoA. So we're going to combine the acetyl group with CoA, and we're going to make acetyl-CoA. At the same time, we need to regenerate our lipoamine. But we can't because there's no disulfide bond there. What do we have to do to that disulfide, to those 2 sulfurs, in order to make a disulfide bond? We need to remove the hydrogens, right? So how are we going to remove the hydrogens? We're gonna transfer them. We're gonna do a redox reaction, right? Okay, so we're gonna convert FAD to FADH2. When we do that, we're gonna reduce the bond, and now we have a disulfide bond, right? Now that disulfide bond is ready to go in lipoamide to go again. But now you have FADH2. FADH2 is going to transfer those hydrogens and electrons from FADH2 to NAD+, and so from this we're going to produce 1 molecule of NADH. And we're going to make acetyl-CoA. Now we're ready for the citrate cycle. That make sense? A little bit? Yes, you pulled 2 hydrogens here. They moved over here. And now you have 1 on the NADH and 1 here. So yes, yes. Yes, the FADH2 is the reductant. Well, yes, yes. It depends on which reaction you're talking about. Yes. Correct. Correct. Okay. So you have to think about how this is going to be regulated too. When do you want all of this to happen? This happens in one big protein complex. When do you want this to happen? When you need it, when you need energy, right? So when you have high levels of ADP, you want this to go, right? What if you have a lot of ATP? You don't want it to go. So it's regulated by levels of ATP. These are all negative regulators of pyruvate dehydrogenase. We can phosphorylate the enzyme and we can shut it down that way, right? That's an allosteric mechanism. If we have a lot of NADH, that'll actually outcompete the NAD+, right? If you've got a lot of NADH, that's kind of indicative of a high energy cell. Same thing, if you have a high levels of ATP, you have high energy, right? What do you want to happen now? If you're not going to go through citrate cycle, what's going to happen? You need to go storage, right? You don't need to increase your blood sugar levels. Yep, so you're going to go through fatty acid storage, right? And acetyl-CoA will actually also inhibit it, right? And that happens a lot of times where you see the product of one enzyme is its own inhibitor. The more you make of it, the more the enzyme itself becomes inhibited, right? That's a very common mechanism that you're going to see.
The more inhibited, the more.
The less activity it has. The more inhibited, the less activity. So what could some positive regulators be? ADP. What else? NAD+, right? CoA, right? So I'm picking all the opposite things, right? And then dephosphorylation. Anybody know how it becomes dephosphorylated? This happens a lot in muscles. What's the trigger for muscle contraction? Calcium. So if you have high levels of calcium, that will dephosphorylate. It actually activates an enzyme that dephosphorylates this enzyme. So high levels of Ca2+ will, in effect, promote activity of this enzyme. It's a positive regulator. Yeah. So all of these here are positive regulators. So they're just basically the opposite. And will promote. Yep. Promote enzyme activity. So y'all have heard of arsenic, right? Arsenic.
I mean, I know
It's a potent poison, right?
I also know it's one of the things they use to describe the Grinch.
It's true. They did. They did. It was used a long time ago to kill off kings and queens, right? Arsenic poisoning. They think it was actually used possibly to kill Napoleon Bonaparte. Y'all know that? It's a little history lesson today.
But I thought he died, in prison.
Well, how did he die?
I don't know, I just think.
So it's actually prevalent in today's time as well. There are some drinking wells in certain countries, like Bangladesh and India. It happened a lot in the 1990s, where a lot of these drinking waters were contaminated with arsenic, and it's very, very dangerous. And the reason is that it inhibits the pyruvate dehydrogenase complex, right? So normally we have this disulfide bond, right? And we can transfer our group to our lipoamine, right? But what happens with arsenic is that it creates new bonds with these sulfides right here. And so now you can't regenerate that disulfide bond. And if you can't regenerate that disulfide bond, you're no longer going to be able to transfer those two carbons, and you shut down. You've just shut it down completely.
So the reason for that is because instead of ATP or the other like pyruvate that we need in order for it, instead binds to this sulfate.
Yep, it binds to these sulfates. Instead of having that acyl group, right? Then you have the arsenic binding. And so you're going to inhibit your pyruvate dehydrogenase complex, the entire thing. So you're not going to go through the citrate cycle. You're not going to go through oxidative phosphorylation, and you will be in a world of trouble. Okay. All right.
Is it sulfur or acyl?
Say again. The arsenic binds to the two sulfurs right here.
So the sulfur in the body or the sulfur part of the body?
No, the sulfur in the lipoamide. These two sulfurs are in the lipoamide. This is the arsenic. Okay. All right, so let's talk about the citrate cycle. What have we generated so far when we go from pyruvate? We want to go from pyruvate into our endpoints, which we'll get there, but what have we produced so far? We've produced acetyl-CoA. I can't write over that. Acetyl-CoA. What else did we make? NADH, right? We made an electron carrier. So when we look at this, you have to remember what's actually part of the citrate cycle and what's not. So if we look at the citrate cycle itself, you're only going to make 3 NADH. But when we add these up, we're going to get a total of 4 NADH. And a lot of times people kind of forget about that one that came from pyruvate dehydrogenase, and I don't want y'all to forget that. You're also within the citrate cycle, so this is citrate, right? We're going to make 1 FAD. Yes, ma'am?
So when we make one turn, it's not 3.
When you make one turn, it's 3. But when you convert pyruvate to acetyl-CoA, that's 1. And in order to,
It's just an adding of everything all together.
yeah. So if you start at glycolysis and you track all the way through citrate, you can't forget that extra NADH, right? Okay, so everything that we're going to get in order to convert from pyruvate, we're eventually going to get to just molecules of CO2, right? That's basically what we want to do. We want to chop up this carbon-containing compound. We're going to have CO2 as our waste product, and then we're going to have these energy-carrying molecules that are going to go on and produce ATPs. So it's an aerobic pathway. That means we need what? We need oxygen, we need O2, right? Why do we need oxygen? To oxidize, so to oxidize the NADH, and what else did we just list here? FADH2, right? Because we said that O2, right, is the molecule that wants those electrons the most. So that's going to be our final electron acceptor. So we have to have something, like we have it in NADH, we have those enzymes, there's electrons in NADH and FADH2. Where are they going to go? They're going to go to oxygen. And when they go to oxygen, we're going to make ATP. Okay. It's a continuous cyclic pathway, right? We're going to see that our first reactant is going to be our last product, right? That's what makes it cyclic. And we'll see 8 different reactions. And there are some that you should be able to recognize what's important in the different reactions. For example, we have 4 reactions, right, that are making what? How do we join NADH and FADH2 together? How do we name those together? Electron carrier molecules. I wish I could change the width of the pen. It looks so thick.
So do we need to memorize all these, or do we?
You should know them all. Yes, you should know them all. You should. So you have some that make energy carrying molecules. You have one reaction that's going to make a GTP, and really pretty immediately that GTP is going to get converted to ATP. So you can almost kind of think of it as ATP. Yes, that's a substrate level phosphorylation. Yes, it is. Yes, it is. So what else do you notice that might be in a different color that you think might be important in this table?
Citrate synthase, isocitrate, dehydrogenase, and acetyl-CoA.
Look at this. Tell me about these values. They're favorable, they're spontaneous, and they are irreversible. Right, we had some of these in glycolysis. No. So what you'll notice in these tables is you have the standard Gibbs free energy, you don't have the actual. And the reason for that is it's really, really hard to get data from inside of the mitochondria without having contamination from the cytosol. So we can guess what the actual values are, but we don't know for sure at this point. But we think they're more favorable than what's listed here. Spontaneous, favorable, spontaneous, and irreversible. So these are our key regulators, right? If you know that these are irreversible reactions, if you can control these reactions, you control the pathway. What do you notice about all the other values except for one? For delta G?
They're all negative.
They're not all negative, but they're all pretty close to 0. So they're at equilibrium, basically, except for our very last one. Our very last one is positive 29.3. That's kind of high. But here's how the cell makes this happen, is that this substrate, this product does not sit around for long. So the cell immediately uses it in something. And so if you use it up, what's going to happen? You're going to have a shift to the products, right? Shift to products. And then that's going to make the reaction go. So you can overcome that positive delta G value, right? Okay, so this is our entire citrate cycle. So we're going to take each reaction one by one. It's not as bad as you think. It's really not as bad as you think, right? You just kind of got to keep your eye on our 2 carbons, right? This was our we have our pyruvate, right? And we converted it with pyruvate dehydrogenase to acetyl-CoA. So I have 2 carbons that are important. My other carbon, what happened to my other carbon that was right here? This carbon got released as carbon dioxide, right? So now I'm left with this 2 carbon chain. That's my acetyl group. So what we're going to do is we're basically going to track what happens to those 2 carbons as we go all the way around. And in the cycle, how many carbons are we going to lose? If we're going to introduce 2, how many do you think we're going to lose? We're going to lose 2. Look here, 1, 2. And then we're going to end up eventually back with what we started with. Okay? So let's take it one at a time, and then we'll come back to this big diagram. Okay, there's a really nice animation. Can y'all access? Y'all can access these because I linked them. I linked them on Moodle. Yeah, if you have the digital book, you can do it. But even if you didn't buy the digital book, I have the animation linked in Moodle. So definitely go and see. But what I want to do is I want to go through all the stages, because this animation really focuses even more on converting it to acetyl-CoA. And we already talked about that. But it's a good animation to go back to. All right, so reaction 1 is citrate synthase. What are we going to do? Don't even look at the reaction. Look at the word. We're going to make citrate. We're going to make citrate. Y'all are awesome. And we know that's why we call it the citrate cycle, because this is our first product. So it's actually a condensation reaction. And it's very, very, very favorable, right? So this one, because it's very favorable, we're going to call it irreversible. But this is really our rate limiting step of the entire cycle. It is our only, you can only have one rate limiting step. It's by definition, right? Like you got a bunch of people together and you like one person holds you up, right? That's this reaction. All the irreversible ones are places where we can control the cycle, yes, but one step is the slowest step, and that's the rate limiting step. So this is the slowest step. Y'all do mechanisms? Yeah, okay. All right, so we're starting out with oxaloacetate. This is going to be a product at the very end. It's our reactant to start with, but it's going to be our product at the very end. We have our acetyl-CoA. So these are the 2 carbons that originally came from pyruvate, right? We lost 1 to carbon dioxide, we got 2 left. Those get attached to oxaloacetate and we release our coenzyme A, right? This is citrate. Okay. Now, we're gonna have to rearrange our citrate in order to get the right bonds. So our second reaction is just an isomerization. So a aconitase is the enzyme that facilitates that isomerization. And a aconitase has a collection of 4 irons and 4 sulfurs inside of the coenzyme, and that facilitates orientation of the intermediate. So this is our intermediate right here, our cis-aconitate. So what's happening in this first half right here? So we're going to form a double bond. So these are the 2 carbons we're looking at in citrate. So these 2. So what happens is we're actually going to lose a hydroxyl, right? If we lose our hydroxyl, then what happens? And what else do you have? You really have, if you want to write it out, you really have this. So you're going to lose a hydroxyl and a hydrogen, and you're going to release what? Water. So now you have a double bond between the 2 carbons. Now this is cis-aconitate, because see how these are on the same side of the double bond? That's cis. So now what's going to happen is we're going to add water back, and we're going to put a hydroxyl on the other carbon and put a hydrogen on the other carbon. So basically we just flip-flopped them. But you can't really do that. So you have to go through this process. So it's a dehydration. So this is dehydration. And this is hydration.
So for the second step, it accepts water, it absorbs water.
That's no, it produces water. Dehydration
No, no, no,
produces.
like first it creates, well, first it releases H2O, then it absorbs the H2O.
Correct.
Okay, thanks. That's what I.
Correct. So now we've made isocitrate, right? An isomerization, iso, it's just a rearrangement. Now that we have it rearranged, right? Now, isocitrate dehydrogenase, this is an important one. This is another one that was on your list that had a very high negative delta G. So what could you tell me about this one?
It's irreversible.
It's irreversible. So this is oxidative decarboxylation. What does a decarboxylation mean? We're going to lose carbon. So look at isocitrate. It's a two-step reaction, right? First, we're going to have an oxidation. So this is an oxidation, right? And in that oxidation reaction, we're going to convert from NAD plus to NADH, right? That's our oxidation reaction. Now, what happens? They kind of highlight the carbons that you should kind of be paying attention to. So what's happening when we go from oxalosuccinate to alpha ketoglutarate?
We lose the C double O and add extra H2.
So this is really our decarboxylation. So we've really lost our carbon. So this is where our carbon was. And this is really a transient molecule. It doesn't hang out very long. So this step is highly regulated by ATP levels, by calcium, by NADH levels, right? If you have a lot of NADH, this reaction isn't going to go. A lot of them are. We're going to look at them. We're going to look at an overall pathway and look at all the things, but a lot of them are regulated by ATP. A lot of them are regulated by NADH. So yes. And yes, and you got to think that's a really good thing. So the question is, are the irreversible reactions all kind of regulated by the same thing? And a lot of them, yes, like ATP, NADH, that kind of stuff, because what you want to do, if this is a cyclic pathway, basically what you want to do is halt it all the way around, right? You don't want to go through some intermediates and waste energy doing that when you don't need to do that. So if you can basically stop it at multiple points because it's cyclic, that's very beneficial. But then you're going to have individual things that inhibit certain reactions, like the product inhibiting the enzyme, right? And that's going to be specific for the individual reactions. And we'll look at all that together at the end. Don't worry. We're going to look at all that together. And the other kind of important thing to note is remember how we said all of these pathways are kind of connected, right? Well, alpha ketoglutarate is a place where we have a connection. And so I kind of want to point it out to y'all. Alpha ketoglutarate is a shared metabolite when we talk about amino acid synthesis. So it can be a substrate and it can be a product. It just depends on which pathway we're talking about. So a lot of times, if you don't need a lot of energy, like you have a lot of energy, right? Well, you have a lot of energy, that's great. How about we build some proteins? How about we build things that we need, right? And so you can shunt these different substrates within the pathway off to different areas to do different things, right? So, and then you can bring them all back. And I'll show you how all that works. Okay. So let's talk about the next one. Okay, so now we have alpha ketoglutarate. We are going to undergo a dehydrogenase reaction. Remember how we said there was E1, E2 in pyruvate dehydrogenase? It's the same thing. So it's a multi-subunit enzyme and it undergoes oxidative decarboxylation. Same thing. Produces NADH. Same thing. So this one is also irreversible. And so, like y'all were saying before, this is going to also be regulated by the energy charge of the cell, right? Just like pyruvate dehydrogenase. Yes. Yes. Because NADH is a product, it can be negatively regulated by NADH levels. It can also be positively regulated by NAD plus levels, right? There's a lot of overlap in this stuff. So the way to study is to look for that overlap because that's going to help you out a lot. Okay. All right. So we go from alpha ketoglutarate. Now we're at succinyl-CoA. Well, if this is a dehydrogenase reaction and it's E1, E2, E3, what's going to be in that complex? Coenzyme A, right? So when you see alpha ketoglutarate dehydrogenase, I want you to kind of be thinking, oh, that's very similar to pyruvate dehydrogenase, but my substrate in this case is alpha ketoglutarate. It's in the name. Mm-hmm. You don't have to memorize the structure of the molecule. You have to be able to look at them and know like what's happening and at what point are you, but you're not going to have to draw them. You are not going to have to draw them. I'm not going to say draw all of the substrates and label all the enzymes. I'm not going to do that. Yeah. What do you have to do? If you can kind of plot the pathway, what's going to have to happen in order to make the citrate cycle go, right? That's the important parts. Yeah. Okay. So we got succinyl-CoA. All right. So now what's going to happen with succinyl-CoA? Well, we're going to convert succinyl-CoA. We're going to get rid of the CoA, right? Because this is a high energy bond, and we're going to make succinate. Because this is a high energy bond, ooh, it's got lots of energy, right? We're going to be able to use it for substrate level phosphorylation. So this is actually a multi-step sort of thing that's happening, right? The thioester bond that's right here has a pretty high negative delta G value, so negative, about, I think 34 kilojoules per mole, right? Something like that. But putting on an additional phosphate from GDP to GTP requires a lot of energy. It requires a pretty big input of energy, about 30. Hello, 30. Right, so that's enough. Yes, ma'am. That is inorganic phosphate. Well, when the inorganic phosphate is transferred onto GDP, that transfer coincides with the release of succinyl from CoA. So the two things kind of happen simultaneously. So we're going to utilize the energy in this bond here in order to attach an additional phosphate to the GDP to make GTP. So if you look at the overall net free energy of this reaction, it's negative 2.9 for the whole kit and caboodle, right? I don't know how I changed color halfway through there. The whole net reaction from succinyl-CoA to succinate, you get negative 2.9 for delta G, right? Is that the difference between the two? Not quite. It's a little different, right? It's a little different. And so really, you're going to go through several really high energy intermediates in order to go from succinyl-CoA to succinate. It's not a one reaction mechanism. It's many high energy intermediates. And it's like the book calls it pass the hot potato in order to get you to the end point. So we call it reaction 5, but you go through lots of intermediates to get there, okay? And then once you have GTP, then inside of the cell, you're gonna convert the GTP to ATP, right? So once you have, let me extend this page. Once you have GTP, right, you're gonna bring in ADP and you're gonna produce ATP plus GDP, and you're gonna regenerate that GDP.
So after GTP, you bring in ADP in order to become ATP and GDP.
Correct.
Wait, is that G or is that delta? No,
G, GDP, GDP. Okay. All right. Reaction 6, we're getting close. Reaction 6, we got succinate dehydrogenase. Succinate I'm sorry, what? Succinyl-CoA synthase. Ooh, syntheas synthase. Sorry. So that's the one we just covered, reaction 5, right? Okay, so we produced our GTP, but now we've got to get to the point where we can regenerate oxaloacetate, right? Or this is never going to go again. And so we go through one more dehydrogenase reaction here. If you undergo a dehydrogenase reaction, you're going to transfer some electrons, right? That should be like one of those things that automatically links in your mind. So we're going to make FADH2 when we convert from succinate to fumarate. Now this is a really cool reaction because this is where we're going to directly link to oxidative phosphorylation and the electron transport chain. Because this enzyme succinate dehydrogenase is actually embedded in the mitochondrial inner membrane. And so these electron carriers here, FADH2, are going to actually send their electrons over, and I'm not going to write it down because you could say this is the link to the electron transport chain, but you don't have to know the electron transport chain yet. But the FADH2 is going to transfer its electrons over to coenzyme Q, which is in the electron transport chain. But you don't have to know that yet. You don't have to know that yet, okay? But I just want you to know this is a link, right? Everything's connected. All right, we're not back to the structure that we want, right? We're not back to oxaloacetate yet. We have 4 carbons, but we're not in the right structure yet. So we're going to go through 2 more reactions. This one's fumarase. Fumarase is going to hydrate our fumarate. Going to get rid of those double bonds. And we're going to make malate. This is interesting because malate is actually an L isomer. Do y'all remember when we said that practically everything in the body is a D isomer, right? Okay, so here's some exceptions, right? We know that this is a chiral carbon, right? You have 4 different things attached to it. So then this chiral carbon, if you look for the hydroxyl, which side is the hydroxyl on? Left side, so it is the L isomer.
So malate is the L isomer.
Malate is the L isomer. Because these aren't chiral carbons. None of these are chiral carbons. Because pretty much everything else in our cell is designed to work with the D isomer. Like stereochemically wise, if you are used to using a D isomer, you can't use an L isomer. You won't make the same connections inside of the enzyme active site, and no reactions will go. So if we ate L-glucose, we would die because we would starve. Because our enzymes aren't designed to handle those configurations. But we are designed to handle L-malate. It's not the only, but it's one of very few. Yeah. All right, so we're almost there. Last one, malate dehydrogenase. And this is our last oxidation. So we're actually going to oxidize the hydroxyl group, right, of malate. We're going to remove a hydrogen here. We're going to remove our hydrogen here. Transfer those hydrogens to NAD. And then we'll have an extra hydrogen ion. And we'll end up with a carbon double bond to an oxygen. And what is this molecule? Oxaloacetate. And this was the first substrate. This was the first substrate. We're gonna find out when we get to oxidative phosphorylation. This one? Yeah, oxidative phosphorylation. It will generate ATPs for us. It will. Well, it will be important in order for us to generate ATPs. Put it that way. Okay, so I want to go back and I want to look at the whole thing real quick. Oh, so when you go back, go and look and kind of highlight these 2 carbons, and you'll see that these 2 carbons go all the way around the cycle. And then you get to oxaloacetate, here are the 2 carbons. These are the 2 carbons that were initially in pyruvate, and you're going to slap 2 more on top of it. And then you're going to lose a carbon here, right, to CO2. And then you're going to lose your carbon down here to CO2. So you lose carbons basically from the bottom of the molecules and you retain them from the top. So in order to lose the ones that enter the cycle, you got to go through more than once. Yeah, it's like stacking. And then you lose it's not quite the 2 N ones, but it's close to it's the N one and then the 3rd one up. All right, so you have section 4 and 5, which are really short, on YouTube, right? And then I posted, I don't think I posted, because people came to my office this morning. I need to post the long clicker questions. I have like 5 of them for y'all. So for the iClicker questions are for like all of chapter 10. So I'll post them when I go back to my office. Okay, that's it. That look good? Questions? Anybody on Zoom got questions?
I was remembering while you were talking.
Okay.
My other question was this.
All right, let me end this. If nobody on the chat has questions, I'm gonna go ahead and end. If you have a question, send me an email. All right. Oh, did everyone vote on a day for lab final? No, everyone has not voted for a day on lab final yet. Please, please go and vote. I believe you guys who are in Dr. Smith's section will be getting the link today.
Okay.
If y'all don't get the link today, please tell me.
I commented a note because I took off on the day that it was listed in the syllabus.
What is it listed in the syllabus
It
as?
was a Thursday. It says prior to my final Thursday, the 19th.
Okay. All right.